Hey everybody, and welcome back to AP Biology. Now, as you already know, we've told you that the curriculum has changed, and so we're going to start off with evolution, okay? And so the second topic, last time we talked about all the evidence for evolution. We brought up all the scientists that you needed to know, and other things like the fossil record. We went over homologous structures. Well, this time we're going to take a look at the evolution of populations, because to understand evolution, you have to understand that it happens at the population level. In fact, populations are the only thing that really evolve. Individuals don't evolve, right? Because once you're born, the genes you get aren't going to change. But if we take a look at the whole gene pool over time, the gene pool slowly starts to change, a population begins to change. Speaking of a population, let's take Homo sapiens, for example. Time Life magazine did something really nifty one year. I'm going back maybe 8 to 9 years. But when they created this photograph, they were trying to say how we were going to evolve into one complete race. Now, this is thousands of years into the future. The new face of America. There's going to be so much gene flow happening that there'll only be really this one sort of race, or there won't be any race. Humans will all look like this. They had said one time that Native Americans, for example, from South America to North America, all pretty much look the same, that people were all very similar. A similar thing is going to happen, the new face of America. This woman is not real. She's every single race put together in a computer-generated program, but still very cool for our evolution talk. Okay, and then another thing I was trying to show you here is that the seashells, or these snails, that no two are exactly the same, but we'll take a closer look at it in this picture. So, for example, this is what I was really trying to get at. You can see the diversity in the population right there. One thing about sexually reproducing populations is no two individuals are exactly alike. The same goes to zebra. If you think two zebra are identical, you're wrong. These look like stripes. Okay, more like fingerprints, and they're very, very detailed. In addition, a population is defined as a group of organisms that live in the same area that are able to interbreed viable, healthy, right, fertile offspring. Another thing, okay, the variation, okay, so what we're looking at is a geographic variation. We've set up maps where, for example, we're just looking at a particular species of snake, and we've started to map where we would find these slight differences within the population. Over time, we'll be able to measure the evolution of these populations and watch even speciation that we're going to talk about in the next lecture. Okay, but talking about the gene pool, this is a collection of all the alleles in a population. Now, we haven't talked about it yet, but an allele basically is a different form of a gene, right? A gene would have two forms, a dominant allele and a recessive allele. And so that's all we're going to really focus on here. So here we're taking the gene pool of these frogs, for instance. Looks like there's 3 different traits, blue, green, and red, and then you'd put them all here in a circle, right? Or let's say even better, I'd rather use real alleles, where we're saying maybe I'm not sure what the recessive allele would be, but let's say it's a different trait. So here we have a homozygous dominant, I have a heterozygous, I got a homozygous recessive. All the individuals in this particular population, no matter how small, we'll put it all in a gene pool, and then we'll count the alleles up. And we're going to use a mathematical formula to maybe see if evolution has even taken place. Okay, so that's Hardy-Weinberg that we're describing here. The Hardy-Weinberg equilibrium is set out to measure whether or not a population has evolved over a given time. So for example, right, this is going to represent the non-evolving population. These are the 5 points that every AP Bio student needs to know. The Hardy-Weinberg equilibrium has to have a large population, because large populations tend to not evolve as quickly as small ones. Small populations have smaller gene pools, so very small changes will lead to like big evolution. But if you take a really big, large gene pool, okay, when there's only one change, it makes no real difference, no significant difference. And so the large population will not evolve, or be very slow at evolving. No migration. Every time migration takes place, or as we said before, gene flow, you're bringing in new alleles and old alleles are leaving. This brings in a lot of different changes. This can lead to evolution. You don't want your population to evolve. You want to have them isolated. You want there to be no migration. So being on a small island is a great example. No mutations. Now, of course, you can't control this because what mutations tend to do is not give you predictable offspring, but instead throw you a curveball. In theory, this needs to work without having any mutations. No natural selection basically means there's going to be no competition. There are no selecting agents that are going to drive this population one way or the other. In fact, a very stable environment is what you want, and you can say that for that as well. Finally, random mating. Okay, now this is where we allow every single member of the population to mate. The weakest members can mate, and the strongest members can mate equally. Okay, not what you see with natural selection. In natural selection, only the strongest members get to mate, or the fastest members, or whatever it is, the most fit. That's not the case. Every member has an equal chance of mating, and that's why the mating is random. Okay, you got to know the equation. There's two equations I got you to know. P plus Q is equal to 1. And then if we just square both sides of the equation, I'm going to square this side. This remains as 1. You open up this factoring equation. Yes, P squared plus 2PQ gives us Q squared. All right, and everything is listed for you over here. P is going to be the percentage of the dominant allele. Q is the percentage of the recessive allele. P squared is the homozygous dominant. 2PQ is the heterozygous. And really what it should be is 2PQ, should be PQ or QP. Either way is correct. That's why there's a 2 there, okay, because there's 2 versions of it. And then finally, Q squared is the homozygous recessive. So I'd like to just take a look at a problem with you very quickly. And let's say they throw this out. This is a typical AP problem that we've seen before. 200 guinea pigs, 32 of them have white coats. What percentage of the population will be heterozygous brown? Kids see this, they start getting thrown off right away. They tell us that 32 are white, and white is going to be the recessive genotype in this case, okay? If the problem didn't say it, I apologize. Then the first thing I want to do is I want to take 32 divided by 100. When I do that, I get 16 over 100. 32 divided by 200, 16 over 100 or 0.16, 16%. At the same time, okay, all I have to do now is if I have the Q squared as 0.16, I just have to take the square root of that, and then I'll get 0.4. Knowing that P plus Q is equal to 1, if Q is 0.4, then P must be 0.6. Now, if I'm looking for the heterozygotes, I need the 2PQ. So I'm going to plug in 2 times 0.6 times 0.4. Okay, and when we do that, 2 times 0.6 times 0.4 gives us 0.48 or 48%. In this case, heterozygous brown will be 48%. I got to give you a tip. For every single one of these Hardy-Weinberg equation problems, you always have to do 2 steps. This is what stops the kids from getting past a certain point. You need to use P plus Q is equal to 1, and you have to use P squared plus 2PQ plus Q squared is equal to 1. If you're not using both equations, you will get stuck. Please don't get stuck. The other thing is they should all pretty much divide out very evenly, because on the AP Bio exam, you are not allowed a calculator, as far as I know. Again, we said that in the first lecture. In addition, everything should be really basic math that you can work out by hand, okay? And so it's going to be a number that you can take the square root of. And usually they start off by giving you the recessive, but not always. It could be the heterozygote or the homozygote dominant. Okay, genetic drift. This is the second big topic in this lecture. Okay, so genetic drift is when there's a drastic change in the gene pool. For some reason, it could either be bottleneck, where it's a very big catastrophe, or it could be the founder effect, where a few members of the population or the gene pool move to a new area and start their own population. So first is bottleneck. You've probably seen this picture before. Take a look at the representation of all the alleles in the gene pool. This gene pool got dramatically reduced. Okay, remember, genetic drift is when the gene pool gets dramatically reduced, and that's what's happening here. So in the cup is all that's left, the surviving population. Notice we lost completely the yellow allele. In fact, the white alleles seem to be pretty big here, right? And now all of a sudden it's not dominating. All right, this is what happens in the bottleneck effect. One example is what they did to the elephant seals. I'm going back 20, 25 years ago, where they were overhunted. The few members that were left, they passed laws, don't hunt them, and now they repopulated. The only thing is, that increased the inbreeding. That reduced the number of heterozygotes. Every time we have genetic drift, we reduce the heterozygotes and we increase the homozygotes. That's not cool because there's a heterozygote advantage to a lot of traits that we're going to see in genetics. Okay, so the founder effect, same thing. Decrease homozygosity, increase decrease heterozygosity, increase homozygosity. In this gene pool here, I want you to note that very few of the members of the population sailed off to a new island. Maybe they were on a log, whatever it is. They had completely separated now. Now they cannot mate with each other. And so this from the original population, this is a poor representation of what we saw. This is only a small percentage of the gene pool, and it's going to start its own population. Gene flow, as we said before, the only thing I really want you to know about this is immigration and emigration. This is where alleles get to go back and forth between the population. This leads to evolution. This is not Hardy-Weinberg. Okay, and then relative fitness. A lot of times I've seen kids get all screwed up on this. Fitness means one thing: how many offspring you make. Darwin pointed this out. The most fit organisms are the ones that reproduce the most healthy, the most viable, the most fertile offspring. That's it. Okay, it doesn't have anything to do with how strong or fast you are, or even how healthy you are. You can be sick. As long as you've had a lot of offspring, you are relatively fit to the rest of the population. Okay, and that's all I care about. How much do you contribute to the next generation? And if you have a lot of grandkids, forget it. You are so evolutionary fit. Okay, now we have the modes of selection. So there's 3 types of selection you got to know for the AP exam. There's going to be this stabilizing selection, right? We have directional selection and diversifying selection. Really simple. Just remember your original population and which way the graph goes. In directional selection, it'll go one or the other way. That's easy to see, but it stays about the same shape. The shape changes when we go to diversifying selection, or also known as disruptive selection, where the 2 extremes of the phenotypes are expressed, and then very little of the mean population. See, these are just some mice. For whatever reason, this mouse is is really the big representative of the population. Very few white and very few dark. But in this diversifying selection or disruptive selection, we start to see the white and the dark, the extremes, being favored more. Now here, with stabilizing selection, we start to rule out these other mice, and really you start to see the mice start to really look the same. The population gets very stable. Every member starts to look very, very similar. Oh, and by the way, we saw this with human birth weight, okay? You don't want certain things. Stabilizing selection actually is a good form of evolution. We want to see the population, especially with babies. I want to see babies anywhere between 7 and 8 pounds. I don't want to see them less because that means they're premature. I don't want to see them more because that could lead to other birth effects and harm the mother. Speaking of which, sexual selection and sexual dimorphism. If I see a male and female, I know it's sexual reproduction. But the thing is, in some populations, the male and the female look completely different. For example, peacocks. That's the peacock, that's the peahen. They look completely different. Hey, peahen, where's your tail? He's got this gorgeous tail. Okay, but when they're very different like this means evolution plays a big part in their mating. And it's true. The peacock has to do like this little dance with his feathers where you can actually see these eyeballs like move around. We saw this at the Bronx Zoo. All right, big difference. I see this with the rooster and the hen. Big difference. But if you take an example like goldfish, we don't see sexual dimorphism there. I can't tell you which is the male and which is the female. They look almost the same. With lions, it's actually very conspicuous as well. I could see that the male has a mane and the female doesn't. Easy to tell the difference. And so this is where we start to talk about 2 types of selection. It could either be intrasexual, which means within the sex, where between the same sex I'd see 2 males, or in some cases 2 females battling over for the mate. Okay, most of the time it's males. Intersexual selection is between the sexes. This is where the females have the right to be choosy to the male. And so this choosiness, believe it or not, is a battle, especially when you think about birds. Like a bird will put together this gorgeous nest. He'll invite the female over. She'll come look at it and she'll be like, I don't want to. And it's like, what? That's intersexual selection. Okay, he can't blame me. So the heterozygote advantage. All right, so this is basically where it's good to be in the middle. And so I was bringing this up before because we're going to see this in genetics. Heterozygosity needs to stay up. You don't want that going down. And the reason why is a lot of traits or genetic disorders are better off in the heterozygote version. I know this sounds crazy, but the homozygote dominant is not good. Neither is the homozygote recessive. Take sickle cell disease, for example, sickle cell anemia. If you have both normal alleles, you're going to die from malaria, which is caused by the Plasmodium protist. Okay, sickle cell disease would be where you get both bad alleles. You don't want that either. You get the sickle cell disease, you have poor circulation, you'll be fatigued, not cool. But if you have one normal and one sickle cell, you get the best of both worlds. You're a heterozygote. Take example, this moose. Okay, at one time, now I tried to get a really old picture of the moose. They used to have really big antlers. So I found this fossil over here. These huge antlers represent the heterozygote advantage. And the reason why is because at one time, this was a good thing to have. But after a while, it wasn't. Why? Because you get neck problems. I'm sure you could beat up any moose. However, you could get stuck in a branch and predators can come and tear you to shreds. If your antlers were too small, nobody wanted to look at you. You couldn't get any mates. But if you were right in the middle, just like the Goldilocks and the Three Bears, okay? When you're right in the middle, that's perfect in nature because you could still escape predators, but you could still attract mates. Okay, just like sickle cell disease. You're still immune to malaria, yet you don't have the full-blown sickle cell anemia. Heterozygotes, that's what we want. And that's what genetic drift takes us away from. Okay, the last thing is why natural selection can't fashion perfect organisms. This is really nice to sum everything up. One, selection can only act on an existing variation. So you can't just have wings if there are no wings, right? It takes a long time to come up with something like this. So whatever variations are existing in the population, that's all evolution is allowed to work on. Evolution is limited by historical constraints. So anything that kind of happened in the past is really going to play a big role in that as well. Adaptations are often compromises. It's true because, you have to compromise. Okay, look, we want to make the antlers bigger and bigger, but wait, it's also harmful to have it too big and so forth. Chance, natural selection, and the environment interact. Chance is really the biggest thing. I mean, because a lot of times we're like, oh, survival of the fittest. He's the most fittest. Hey, if there's a meteor shower, like in this historical constraint, all of a sudden you can lose anybody. A meteor shower or a forest fire doesn't care how fast you are, how strong you are, how big your peacock feathers are. It doesn't care. And so because of that, that's why evolution can become very limited. Okay, guys, so this concludes our lecture number 3. In our next lecture, we're going to talk about speciation, the different types of speciation, and then we're going to go into the history of the Earth. All right, guys, so until next time, I'll see you later. Be safe.